Hello, in this video I will discuss about the structure of polynucleotide chain. Nucleotides are the building blocks of nucleic acids. Each nucleotide monomer has three chemically distinct components: a nitrogenous base, a pentose sugar, and a phosphate group. Nitrogenous bases are of two types: purines like adenine and guanine, and pyrimidines like cytosine, thymine, and uracil. Purines are same for both DNA and RNA. Out of the three pyrimidine, cytosine and thymine is present in DNA, whereas in RNA there is cytosine and instead of thymine it has uracil. The pentose sugar found in a polynucleotide is either ribose or deoxyribose. The difference between these sugar is the presence of hydroxyl group on the 2' carbon of the ribose, which is absent in the 2' carbon of the deoxyribose. A nucleic acid which contains ribose is called ribonucleic acid, or RNA in short, while the one containing deoxyribose is called deoxyribonucleic acid, or DNA in short. Now let us number the carbon atoms of this sugar. Here is the first carbon, second, third, fourth, and this one is the fifth carbon of these pentose sugar. A nitrogenous base is linked to the first carbon of pentose sugar through a N-glycosidic linkage, and this whole unit is called a nucleoside. When the nitrogenous base adenine links with ribose sugar, it is called adenosine. Similarly, when guanine links with ribose sugar, it is guanosine. Cytine with ribose is cytidine. Uracil with ribose sugar is uridine. If the nitrogenous base adenine is linked to deoxyribose, it is called deoxyadenosine. Similarly, if guanine is linked with deoxyribose sugar, it is called deoxyguanosine. Cytosine with deoxyribose sugar is deoxycytidine. Thymine with deoxyribose sugar is deoxythymidine. Now, when a phosphate group is linked to the 5' end of sugar through phosphoester linkage, a nucleotide is formed. Let us have a look at the linkage formed once again. Base is linked to 1' carbon of the sugar through N-glycosidic linkage, and the phosphate is linked to the 5' position through phosphoester linkage. Let us see how nucleotides are joined to each other. Here is our first nucleotide, and to the 3' carbon the phosphate group of the next nucleotide will be attached. And the linkage that is formed between 3' carbon and the phosphate is called phosphodiester linkage. Similarly, to the 3' carbon of this nucleotide, phosphate group of the next nucleotide is added. In such a manner, many nucleotides are joined to one another to form a polynucleotide chain. A polymer thus formed has a free phosphate attached to the 5' carbon at the left side, which is referred to as 5' end of polynucleotide chain. And at the right side, you can see there is a 3' OH group. This is referred to as 3' end of the polynucleotide chain. The sugar that I have used here is deoxyribose. Had this been ribose sugar, there would have been an additional OH group in the 2' position as well. So we can see here that the backbone in a polynucleotide chain is formed due to sugar and phosphate. The nitrogenous bases are linked to the sugar moiety projects out from the backbone. So we have come to the end of the video. Thank you for watching.